our YouTube channel. Okay, that should be okay. All right, so welcome everybody. It is my great personal pleasure to introduce to you Dr. Michael Pereira, who we are hosting today in the framework of our lab's series. Let me give you a short bio of Michael. So Michael earned his master's degree in communication systems from EPFL in Switzerland. He spent 2 years as an engineer in Sony in Germany. Then he returned to EPFL for a PhD with Professor José Millán. So this is when we coincided with Michael while doing our PhDs. During his PhD, he focused on discrete and continuous error monitoring processes in the brain, where he published a lot of interesting articles, if I may add. He completed a first postdoctoral fellowship at EPFL with Olaf Blanke and Rezai from West Virginia University, investigating the neural correlates of perceptual consciousness. He then pursued a second postdoc with Nathan Faivre at the Laboratoire of Psychologie and Neurocognition in Grenoble in France, funded by the Swiss National Science Foundation. Michael is now a scientist at the French National Institute of Health and Medical Research, INSERM, based in Grenoble, in Grenoble's Institute of Neurosciences, where he leads the modeling and estimation of subjective states team. His research explores the neural mechanisms underlying conscious experience through computational modeling and single neuron recordings in humans. He aims to translate this work to clinical settings, studying hallucinations and psychiatric symptoms, such as pathological doubt in obsessive-compulsive disorder, with which most of us suffer, I would say. So that's very interesting. So the title of Michael's talk today is exploring subjective aspects of perceptual decision-making with intracranial recordings and computational modeling. So without further delay, Michael, feel free to share your slides and the floor is yours.
Thanks. Well, you should see the slides, right?
Yeah, it's already there, yeah.
Okay, and now the important thing is, do you see my mouse cursor? Yes, okay, cool. So hello everyone, it's a pleasure to be here virtually. Thanks for inviting me. And so I'm going to talk about these subjective aspects of perceptual decision making. So we're a group of 8 or 9 people, depends on how you count in Grenoble, who have contributed to this work I'm going to present. And so I talk about subjective states or subjective aspects, but to be honest, it's just a way to hide what we kind of call the C word, which is like trying to get as close as possible to neural correlates of consciousness. And so, yeah, the problem is when we talk about consciousness, then we get these images on Google Image. Oops. And so actually, when we tell people about the work that we're doing on consciousness, it gets pretty boring. So because we just show faces to people and ask them whether they perceive it or not. And so in this talk, I'm going to kind of hover between perceptual decision making and consciousness and trying to get as close as possible to this subjective experience we have of, let's say, the outside world. So in consciousness science, there are like 2 big, somehow orthogonal, but dimensions. So you have levels of consciousness, which vary from coma to conscious wakefulness. And depending on which level of consciousness you are in, you might experience different contents of consciousness, which is really like the content of what your subjective experience is. And so in this talk, I'll just be talking about stuff that we do during wakefulness. And the way we try to approach this subjective experience that we have when we see something or when we hear something is through detection tasks. And so you imagine that you subject participants to like a very a very weak vibrotactile stimulation on the wrist. So in some cases, the participants will perceive it. So they'll have a subjective experience of this vibrotactile stimulation, and you'll have some activity in the brain. And since the vibrotactile stimulation is so weak, well, in some other cases, for just like exactly the same intensity, they will not perceive it. So we talk about a mistrial. And in that case, they don't have any subjective experience. So that we use this tactile stimulation in paradigms where you have, for example, a start cue, then there is some time where they could or could not be a stimulus. And at the end, we ask them to respond. And we compare the brain activity between when they report perceiving the stimulus and when they report not perceiving the stimulus. So that's the contrastive approach. And ideally, that should isolate correlates of this subjective experience, since the sensory stimulation is always constant. The problem is when you look at it, for example, at the scalp level with EEG, you see some differences between trials where people report perceiving a stimulus and trials where they don't. But this is actually, and this we hope is related to this perceptual experience, which is brain activity that's present or absent when there's a perceptual experience or not. The problem is that there could also be other types of cognitive processes that confound this activity. So you could imagine that if your attention is high, then you're going to perceive it with a higher probability than if your attention is low, and that will contaminate these neural correlates. At the same time, you could imagine, well, you ask actually participants to report what is the content of their perceptual experience, and so there's a part of all this that is related to the decision-making process that leads to the report or not of the stimulus. And so that's kind of why it's interesting to go to intracranial recordings, so that we really get the granularity to manage to pinpoint this activity related to perceptual experience. And so during the whole talk, we'll be trying to, with more or less success, to fight against these either prerequisites of consciousness or perceptual experience, or these consequences like decision-making. And so if you look at the whole spectrum between sensory regions that are processing sensory information, and these are always activated no matter the let's say the perception that comes out of this. So you could imagine, for example, the retina or the early visual cortex, and this the level of activity is going to covariate with the sensory the stimulation intensity. And at the end, you have the choice of the participants, so whether they report the stimulus or not. And obviously, these correlates of subjective experience, they're not going to be in either of these two extremes. They're going to be somewhere in the middle of this whole hierarchy of sensory to choice processing. And well, we have pretty good techniques to be sure that it's not like purely sensory neural activity that we're looking at, or purely choice activity. But then everything that's in the middle and that I'm going to talk about in this talk is possibly contaminated, as I was saying, with these decision making, so consequences or prerequisites. And so the whole goal of the field is to find neural correlates of consciousness, so which are defined as minimally jointly sufficient brain processes for a conscious perception to occur. And using computational modeling, we'll be also interested in trying to reproduce what could be the underlying mechanism. So using this contrastive approach, so contrasting trials where the stimulus is perceived and trials where the stimulus is not perceived. So there've been a lot of work on this. So you can see a lot of differences in the fMRI activity, in EEG activity, or in intracranial activity. Not so much actually using single neurons recordings. So this is an example from Quiroga's work where you see a neuron in the medial temporal lobe that has a different firing rate for recognized versus non-recognized trials. But there are only like less than 10 studies looking at this single neuron in the human brain. And none of these studies look at what's happening under the cortex, so in subcortical structures. So we have a lot of theories that involve the thalamus and a little bit the basal ganglia, but we've never really managed to go and record neurons down there. So the first study I'm going to talk about, that was done in collaboration with the EPFL and the West Virginia University in the US. We had the chance to be able to go to surgeries where they implant deep brain stimulation electrodes. And so what happens during these surgeries is that the patients arrive, they're fully anesthetized. Meanwhile, the neurosurgeon is going to drill the hole in the skull and insert a microelectrode. And then the patient is awoken from anesthesia because the clinical team wants to record the neurons along the track leading to the deep brain structure that is targeted. So here I'm giving you an example of a track that's going down to the I think it's the thalamus. Yes, it's the thalamus. And so in the last, on the last bit of this electrode trajectory, you can see the signal that is recorded, and you can see the spikes of single neurons that you can record and actually listen to know exactly in which brain structure the electrode is. So that's done by the electrophysiologist. And at that moment, we had like 10 minutes to do an actual cognitive task with the patients. And so the challenge here was to manage to find the intensity of these vibrotactile stimulation that was weak enough for the patient to only feel half of the stimuli. And so we had to throw away a lot of data. But at the end, we managed to get some neurons that were responsive. So it's again the same task I was talking about. So you have a start cue, then you have a 2 seconds window where the stimulus could come at any moment, and then we add the delay, and finally we ask the patient to respond whether they felt the stimulus or not. And so looking at the signal, we can isolate the action potentials of the neurons, and we can try to cluster them into, let's say, clusters of individual neurons, and then compute the firing rate over time. And so here in this green box, you see 2 examples of firing rates of 2 different neurons across time. It's time locked to the onset of the vibrotactile stimulation. And you see this difference of the firing rate related to the baseline. So these were like probably purely sensory neurons that just changed their firing rate after the stimulus. And then we found another, let's say, family of neurons where the firing rate actually was different when the patients reported feeling the stimulus, so for hits, compared to misses when they reported not feeling the stimulus. And that's the kind of activity we were looking for. But as you can see, the weird thing was that the activity for missed stimuli was higher than for the hits in quite a lot of neurons. And that might be because of the way that or at least that's what we speculated, that's because of the way that the basal ganglia is wired, and that the subthalamic nucleus has an inhibitory role over the cortex, so it might be just filtering the sensory information that reaches the cortex. And this is, of course, very speculative. And so to close this this chapter, we looked at the different timings of these effects. And although we had like very few neurons, because we collected like between 0 and 3 neurons per surgery, we found that there was quite bimodal distribution of these effects, with the first, let's say, cluster of, or time cluster between 100 and 200 milliseconds after the onset of the stimulus, and the second cluster around 300 milliseconds. So which could suggest that you have sort of two waves of processing in these different structures. So what I didn't say also is that the two structures that we targeted were the thalamus and the subthalamic nucleus. So that's for the first study. So we get some very nice opportunities to record neurons in the clinic, but of course we have to adapt the research that we're doing to the clinical settings and to the length of the surgery. So it gets pretty difficult to get some really, how could I say, clean and massive amount of data. We also had some another opportunity to record single neurons in humans. That was again with EPFL and the University of Geneva. And there we had one patient with intractable epilepsy that came to the hospital to receive an ECOG grid that would allow the clinical team to monitor the epilepsy for a week. And within a clinical trial to study epilepsy, this patient accepted to receive a microelectrode array that was implanted in just next to the postcentral sulcus. So in the posterior parietal cortex, but just next to the somatosensory cortex. And so these UTA arrays, they have 96 microelectrodes, and with a bit of luck, each of these microelectrodes is able to capture the activity of a few neurons. And so we had actually the same task, except that this time, so vibrotactile stimulation, sometimes the patient felt it, sometimes the patient didn't feel it. And there we had different versions of the tasks. So in this first experiment I'm going to show you, we asked the patient to press a button as soon as they felt the stimulation. And so we can separate the firing rate of the neurons that we record based on the reaction time of the patient. And so you can see here, this is for the fastest reaction time. You have a quite steep increase in the firing rate before the button press. And then if you take a slightly slower response time, you have a similar increase, but that is with a slope that is a bit smaller. And finally, for the slower response time, you have a much less steep slope. And when the patient doesn't report the stimulus, well, the firing rate stays pretty flat. And so this is a sub-selection of the neurons that we could record, but we were quite excited about this because it very much looks like the neurons related to evidence accumulation that were found in non-human primates studies. We did some other tests. So for example, we averaged the firing rate of all these neurons, and we looked at the single trials ordered by the response time here. So the black part is hiding the part after the response times. And you can see that the increase starts at a rather similar moment and reaches a certain level just before the response. And on every single trial, the slope seems to correlate negatively with the response time. So we also had a computational model with just a few parameters that could actually reproduce qualitatively the firing rates that we obtained. And so here we have a difference between stimuli that are let's say reported, perceived, and stimuli that are not reported. But of course, there's a major confound if we want to link this to the subjective experience, because on the blue curve, there's a button press, and in the red curve, there is no button press, right? So we had the second experiment where this time we used the same design. So we waited for delay before asking the participant to respond vocally on whether they detected the stimulus or not. And then we also asked them for the confidence they had in the detection. And here, if we look at the neurons that are responsive to hits and misses, there is a pretty small difference in the firing rates between hits and misses. So we could start relating to the subjective experience of the stimulus. But then if you separate these blue curves into trials where they reported having a high confidence and trials where they reported having a low confidence, you'd see that there is a major difference. And so it seems that, let's say, not only was the firing rate modulated by the detection, but additionally by the confidence that the patients had in their response. Here you can see the example of a single neuron. So on the lower panel, you see the increase in firing rate for high confidence trials compared to low confidence and misses. And here you see a raster plot where like every little dot is the firing of a neuron, and you see the trials ordered by condition in this big increase in the firing rate of this neuron for high confidence hits. So then we asked whether using multivariate decoding, we could still differentiate low confidence trials from misses. So we put all the firing rates of the neurons inside the decoder, and in a cross-validated manner, we looked at the area under the curve for high confidence trials and low confidence trials related, let's say, compared to random permutations. And you see that in the low confidence trial, you're still able to discriminate hits from misses. And the last thing we did with this in this experiment was we wondered how we could decode confidence. So in all the data that I showed you, it's aligned to the stimulus onset. So you have these vibrations on the risk that are all aligned. But since they're coming at a different moment, the start of the trial is at various timings before. And when we train decoders on the onset of the stimulus, well, we could quite well decode the 3 levels of confidence for when the patient reported the stimulus. But we were not capable of decoding the confidence they had in mistrials. And so what we did is we reused this decoder that was trained on the stim-aligned data. And again, in a cross-validated way, we applied it over the whole window, starting from the cue and just ignoring whether the onset of the stimulus. So here we have no idea of when the stimulus, the decoder has no idea of when the onset of the stimulus occurs. And we found that there's actually, we could still decode the confidence in hits, but we could also decode the confidence in misses. Which, okay, so obviously this is one patient, and we were able to run this experiment only once, so we have to be a bit careful with the interpretation. But it could, if it holds, it could suggest that what's driving your confidence in the absence of a stimulus is not happening exactly or necessarily during the stimulus, but over the whole window where the stimulus could occur. Okay, so let's do a little interim summary. So we have these neural correlates of consciousness using the contrastive approach in subcortical regions at the single neuron level. And we have these neural correlates of consciousness also in single neuron activity in the cortex, so in this posterior parietal region. And in the experiments where we have an immediate response, this looks like evidence accumulation in humans during detection tasks. Sorry, and let me show you this in a little schema. So we think that basically what happens is that you show the stimulus, and depending on the noise, basically this noise information is going to be accumulated. It might not reach the decision threshold, and in this case, it stays unconscious. But it also might reach this decision threshold at various timings and lead to the detection and response times. And it also seems that if you look at let's say, the maximum that is attained by this process, you are able to decode the confidence. So this is nice for detection decisions, but is it really related to subjective experience, right? Because it could be just related to the decision making. And actually, if you look, maybe I'll go fast through this and just show you this this plot. This is EG activity when the stimulus is task relevant, and you see this increase in the activity. But as soon as the stimulus is not task relevant anymore, so you just passively show the stimulus to the participants, well, this activity completely disappears. So it cannot really be related to subjective experience because you're able to perceive the world without necessarily reporting a decision, right? So in the next study, we we didn't go for single neuron recordings, but we used local field potentials that we record in patients that go for epilepsy surgery. And so they're monitored for a week or two in the clinic with these electrodes, and we get a pretty good coverage of some of the regions that are of interest for us. So here we used a visual detection task, but it goes exactly the same way. So you have a face that's embedded in noise. It's very difficult to detect. And well, the participants press a button when they detect the face. And we had some pre-registered brain regions or regions of interest that we were looking at. And so here you see the number of channels for each of these regions. You have the ventral visual cortex here, the superior parietal cortex, inferior frontal cortex, and the LPFC here. And in two of these regions, in the ventral visual cortex and in the inferior frontal cortex, we again find these activity that looks like evidence accumulation, meaning that the slope depends on the response time. So this is short. These are short response time, medium response times, and slow response times. So based on this, we went for okay. So this is the activity, and so in the inferior frontal cortex, so in the and a bit of the insula. And then here is the activity when we introduce a delay. So there we don't have any idea of what's the response time. But we still see an increase in activity, both for the stimuli that are at threshold and the stimuli that are shown with a slightly higher intensity compared to the misses. So that shows that it's not just the button press that is driving this activity, but it's really like at least the decision or the subjective experience. And then we asked, so we just showed the same exactly the same pictures of faces to the patients, but in a passive way. So we didn't ask them to do anything during the task. And so we thought, like, if it's just related to decision making and then not making a decision, then the activity should be pretty flat. If it's related to the subjective experience of the face, then we should see a difference in the activity. You see here that for the insula, there is, well, we could imagine that there's a small increase, but it's basically non-significant. And so we interpret this region as basically related to decision making and not subjective experience. So that might be just a consequence of this consciousness. Then in the ventral visual cortex, so this is the brain region where we think that the most of the face processing actually occurs. You still see this evidence accumulation. This activity is still present when you introduce a delay. And during this no report paradigm, so where we were just passively showing the stimuli, you see activity at the same timing, so it's not very high for the lower intensity, but for the higher intensity, you can clearly see this activity. So which suggests that it's beyond just the decision making. It's not just a correlate of decision making, but it could be related to the subjective experience of seeing a face. And then, so this was the work done by François Stockart in the lab, and there was another PhD student that was more on the computational modeling side who used the model which has just a few parameters, and it's actually implementing mathematically what we think is occurring for evidence accumulation. And it was pretty easy for her to find some parameters that were reproducing quite well the evidence accumulation in experiment one. And then just keeping the exact same parameters of the model, she could also reproduce what was happening in the delayed version and in the no report version. So which gives us a hint, let's say, to support this idea that evidence accumulation might be a mechanism that is underlying the occurrence of your subjective experience. And finally, we went also for decoding approach. So here we designed a new decoder that was actually optimizing weight, but on the whole length of the signal. And so it's getting the optimal weight basically, so that if you would set a threshold to the weighted sum of all channels, you would get the best accuracy to reproduce whether a stimulus is perceived or not. And so in a cross-validated way, we could, in some subjects, let's say, get a significant decoding of whether the trial was a hit or a miss. Here you see for the two most interesting regions, the number of subjects out of the total that were for which this was working. And this was actually highly dependent on the implantation. So we actually saw that the closer we were to the actual coordinates in fMRI studies of the face processing areas, the better was the decoding was. But with this new decoder, the nice thing is that we can also define at what time this weighted sum is crossing the threshold. And so this decoded time actually also correlated with the response times, although the decoder had never seen these response times. And this worked in like all the patients for which it worked for the decoding. And then we applied this decoder out of sample to the other experiment where there was a delay. And we were still able to decode the difference between hits and misses. Here you see an example of this this weighted sum. And finally, we applied again this decoder to decode the intensity in the passive task. Unfortunately, out of these 4 subjects, we only had 2 that managed to do the 3rd experiment, and it worked in 1 out of 2 participants. So that was a bit weak, but still we think that it's basically what we can interpret from this is that you have some neural code or some weighting of these different electrodes for every patient. And it seems to be that the same neural code that is trained on the first experiment is able to decode in the other experiments. Okay, so here the addition is that we still have this evidence accumulation process that in late visual areas such as the fusiform gyrus or the ventral visual cortex I was showing you is also present during passive viewing. And we have this fact that the behavior and the electrophysiology is well described by a simple computational model. So that's kind of it for the part on electrophysiology. But we still have this question of whether this evidence accumulation is it something that is necessary for your subjective experience to occur, meaning that you need this threshold crossing to then start having your conscious perception, or is it constitutive of it, meaning that the whole process is actually what your subjective experience is? And so for now, we're turning to computational modeling, and with the idea that the sensory information that you get in your visual cortex is very noisy. So here you'd see the activity of a stimulus, of course, in the model. And then you have this huge noise above it. And so we think that if this is the case in the brain, then you have to have some smoothing of the signal. So one way to do it would be exponential smoothing, and the more you smooth, the stabler becomes your experience of the world. But of course, you shouldn't be smoothing too much because otherwise everything would get sluggish and weird. And so we think that this is, let's say, the way that sensory evidence is accumulated for subjective experience. And if this is the case, that also means that the higher the intensity of the stimulus, the longer it will take to get down again. And so the longer will be your perceived duration of the stimulus. And sorry, that's actually something that is pretty well shown in the literature on psychophysics for like the last 60 years, but I think, or at least I hope, has not really been shown with the computational model. And so that's the task that we ran. So it's still the same design where you have faces that are difficult to perceive, and participants press on a button as soon as they see the face. And at the end, either we have a confidence rating. So they rate the confidence they have in their detection decision. Or we have what we call duration blocks, where we ask the participants to press a button for as long as they thought that the face was lasting. Then, since it's a computational modeling study, we actually ran healthy participants for 8 consecutive sessions, or like over 3,000 trials, with different levels of intensity of the face. And we had the dynamic condition where the intensity was changing over time, so either ramping down or ramping up. Okay, and so this is work again by Ramlam Sheikh. The model here is, so here you see in the dots are the behavioral data for one of the 4 participants we ran. And these lines are what the model is able to reproduce with only 4 free parameters. So you see that for the detection report, so whether the participants detected the face or not, it works pretty well. Same thing for the response times. So the model reproduced the fact that the higher the intensity, of course, the faster the participants are. And then we turn to these more subjective aspects of the decision. So we tried to model the confidence as the height that this process would reach over time, and the duration as, let's say, the duration of the whole process at a different threshold. And so we kept the model as it was trained for the detection and response time, and we just added 2 additional parameters for the bias and sensitivity, and we were able to reproduce the confidence. And by applying a second threshold, so thinking that let's say you might be a little bit conservative in your responses and you might still perceive something that you don't report, measuring the duration under this perceptual threshold allowed us to reproduce what is called this magnitude duration effect that is pretty well known. So this light blue curve is the short duration stimuli, and this dark blue curve is the long duration stimuli. And of course, participants report longer durations when the duration is longer. But the interesting thing is you have this super strong effect of the intensity of the stimulus. So when you increase the intensity of the stimulus, people report it to last longer. And that we were able to reproduce with this, let's say, very simple model. And so I'm nearly finished. I'm just going to like kind of showcase a few of the like really ongoing studies that we have now. So Artemio Soto Brechera is going to more complex models. So there it's not a simple mathematical formula with 4 parameters, but he's actually modeling recursive neural networks to model the firing rates with the synapses, and he's modeling two regions, so a sensory region and a decision making regions. And then the idea is that we can try and reverse engineer these models to see a bit what's happening inside. And so this is very, very preliminary, but if you look at the data I showed you just before for the modeling study, this is just what we will produce with an untrained biological model. So you see that we do reproduce the increase in detection for long and short stimuli. We do reproduce the shorter response times for increased intensity, although the shape is a little bit different. We reproduce the increase in confidence for increasing intensity, although we have some weird effects here for the ramping up and ramping down stimuli. And finally, we also do reproduce this magnitude duration illusion, where when you increase the intensity, it increases the perceived duration. So this is done just by measuring how long this increase in firing rate in the sensory region lasts for. And the other thing is that if you just look at the firing rates of the model, they actually sort of look like the high gamma activity that we measured in the local field potentials. So okay, so this I actually already showed you at the beginning, but the way we think that we might go to really isolate the activity that's related to perceptual experience is by using the dynamics of this perceptual experience. So while most of the works that have been done until now have focused on whether stimulus is perceived or not, we think it's important to actually also model like how long do you perceive the stimulus, or how intensely you perceive it, which is like measured through confidence ratings. And so now we want to still have this contrastive approach, but we want to add these duration reports, and then always check whether the activity is still present when we are not when it's not task relevant, so during passive stimulation. And so we're checking this dynamical approach right now with another PhD called Shiva Madhyan, and we're replacing basically the electrodes that are used in the clinic by hybrid electrodes that have like the same clinical benefits, but that also have these tetrodes that can be protruded from the shaft of the electrode and that can measure single neuron activity. This is also very preliminary. It's just the firing rates of multi-unit activity that increases for houses, objects, and faces. And now we're going to see whether it can explain the subjective duration reported by the participants. So, well, I hope during this talk I've sort of convinced you that evidence accumulation doesn't only happen like at the latest stage of decision making, but that it also occurs a little bit earlier, that you observe similar activity in the absence of a task. So it must be a bit more than just decision making. So that's the way that we use to deal with these consequences of decision making. And that we have some, let's say, preliminary ideas about what could be the underlying mechanism with these computational models. And then using like these different subjective aspects that are related to this decision-making process, so not only detection, but also confidence and subjective duration, we might be able to isolate any other cognitive process that actually doesn't have the same duration. Okay, and so evidence accumulation or temporal integration seems to be a quite ubiquitous mechanism in perception, but it's kind of rarely considered in relation to consciousness studies. And so if this is true, then it means also that these all these processes that are going to occur at different timings, and this might vary across trials or experimental conditions. So it might not make so much sense to really look for precise timing of when our subjective experience occurs. And in terms of anatomy, it seems that for the visual task, it's most in these late visual regions. But of course, we cannot exclude any other region also, because some regions are not implanted in these patients with epilepsy, and that we will use this dynamical approach to pinpoint this hopefully true neural correlate of consciousness. And then I just want to mention that there are a lot of theories around consciousness, and so there are like some ways to relate this to these theories. Where is my thank you slide? Here, there was a thank you slide. And so
Don't worry.
thank you for your attention. I'd like to thank the patients for doing these experiments at a difficult moment of their lives, collaborators, and well, happy to take some questions.
Thank you very much. It has been really, really interesting. So yeah, we can move to questions. I think the most efficient way is if everybody can use the React icon in Zoom and raise their hands, and then I will point you to start your question. Or if you prefer, you can also write it in the chat, and I'm going to transfer it to Michael. So while people are gathering their arsenal of questions, I have one for you, Michael. So I'm conscious that you really want to study this integration process, and but for us, I believe it is really interesting, or more interesting, at least, to go back to this black and white question about consciousness. So as you know, it is in BCI field is a very active field, trying to discern the level of awareness or consciousness in people with disorders. So I wanted to ask you whether you have any insights on how much your research translates to this area, so to people with disorders of consciousness, and especially if we have to do it with a non-invasive approach, let's say pretty much EEG. So, and I guess there are two legs to this question. First is, do you think the neural correlates that we could get from EEG could deliver the same, accuracy like your intracranial recordings? And the second is how critical is for training these models that you showed to have the ground truth of the response, because obviously in these situations we cannot get a response, right? So yeah, please.
Okay, I'll try not to forget any leg of your question. So yeah, it's an interesting question, and actually we should chat because we're starting a project in Alabama where the goal is to record like invasive data the way we did, but then we play. So we do this in the auditory domain because when patients sleep, of course, we cannot show them faces. But during the whole night, we try to play the same sounds and to see how the levels, or let's say, or how these classifiers react to different levels of sleep. And then when they get explanted, we have like a short window of opportunity to record, to play them the sounds while they're anesthetized. And so with this, we want to try to branch these two aspects. Then for the specific, or let's say for the sensitivity of EG, I really don't know. So like what we did see is that, like these EG correlates, they completely disappear in the absence of a task, while we actually still see this activity. But then I don't know if you would try to kind of use like state-of-the-art reconstruction methods and really target these. Is actually like for vision, it's really like a bad region, I think, to record. It's just like under the temporal lobe. I have no idea what you would be able to get out of this. And the last leg was
whether we can live in this clinical setting with the absence of ground truth that you use in your models through the response, because we cannot get a response. Did you feel or not the stimulus? Somehow only thing we have is the brain response, no matter what modality we have the same. And then we have to make a decision whether this person perceives the stimulus or not.
Well, so we have the same problem, of course, because we also want to record activity, but in the absence of a task. But we have this advantage that is in the same patient, so we can train classifiers and then apply them on these same patients. Now, I have no idea whether transferring this to a completely different individual is feasible or not. I think there are some clinical trials that actually stick electrodes in these patients. It's really a literature that I don't know much.
I guess
I
we'll
guess
have
I
to
would
do
try
some more
to.
experiments. Yeah.
Yeah, I will try to have both modalities at the same time and see how much you can get while knowing, let's say, not the ground truth, but at least the intracranial version of the EG.
Okay, thank you. Is there some other question from the audience? Sebastian, please go ahead.
Hi. So yeah, thanks for the talk. And obviously for practical reasons, you study this with sensory stimulation, but for conscious experience, also experiences that are not based on sensory stimulation play an important role, like dreams or memories or just thoughts. So how does this evidence accumulation model apply there?
So that's Actually, super interesting. It's even tougher, I think. For memory, there has been some, at least behaviorally, there have been some models that actually, I think some of the first models were used in memory, like 40, 50 years ago. So it seems that there might be something similar happening in memory. For thoughts, it's really difficult to like operationalize. We're also interested in this for work that we do in obsessive-compulsive disorder with these unwanted thoughts. We have some colleagues that are doing paradigms with like think, no think tasks where they ask the patients to either think about something or not think about something. You have these kind of Libet clocks where you ask participants to go back to the moment where they had a thought. But yeah, all this is still pretty difficult to achieve. And I have no idea whether they're based on evidence accumulation, but at the same time, the way that we define evidence accumulation is like temporal integration. And when you feed activity to a large neural network, you have sort of automatically some temporal integration. So it seemed to be a good way to go.
Okay, thank you. Is there any other question? I will ask a second one while we're
Yes.
waiting. Yes, please.
Sorry.
Please, Selina, go ahead.
I could not find the
Please go
hand.
ahead.
Okay, thanks for the talk. Very interesting. I was just curious about, did you also take a look at the unconscious processes before it comes to decision? Because a lot of decisions before you can report are made unconsciously. So I know it's from the visual perception, not so from the sensory part, but this would be also interesting to know.
Yeah, absolutely. I think there's a pretty big fight in the community about to what level can we define this unconscious processing. We didn't look at this specifically, but at the same time, like just looking at your level of confidence when you don't detect a stimulus could be seen as some kind of unconscious processing, and we get some kind of mixed results. So I showed you the decoding that was working with this trick of looking at the whole window, but at the same time in the LFP study, we were not able to decode the confidence in the misses in these regions that we were looking at. With the computational model, it works pretty well. What other links could I make to unconscious processing? And this, of course, is for you to say that it's unconscious processing, you should be in the team of people who think that just saying that you didn't see something is sufficient for it to be unconscious. But it could be that you just got a glimpse of it, and that's so then it's not really unconscious processing. And that's also the reason why we want to have like these two layers of recursive neural networks, so that we can have like a different threshold in the sensory region and try to model these trials where you might have just perceived a little bit of the thing, but you're not reporting it.
Okay. Thank you.
Thank you. I think we can have one last question by Umesh Kumar Naik. So please, Naik, go ahead.
Hi, Dr. Michael. It's a really fascinating work that you have presented today. I just do have two couple of questions here. So the first one is something like, how do you quantify this decision threshold and the perception threshold? Does it vary over the subjects, or is it like independent?
Yeah, it varies because the subjects is probably also, we didn't test that, but others did. It probably also varies. The decision threshold is going to vary based on some external factors. So for example, if you if you reward the participants differently, meaning like if you tell them now you really have to detect every possible face, or compared to if you tell them that the important thing is to never never report a false alarm, so never report a face when there was no face. So you have you change between a kind of liberal reporting and conservative reporting in terms of perception, of course. So this decision threshold is going to vary, and this decision threshold is kind of easy to fit because we have the behavioral data of really like what is the decision. And for the perceptual or sensory threshold, we can only guess, and that's why so there's no real way to know where it is. What we do is we use these the subjective duration reports to fit this threshold. That's the best we can come up with. So we hope that basically by using different proxies to this subjective experience, we're going to manage to better get through this latent variable.
And thank you. And the second question that I had is like the purpose of using this decoding network, was it to extract some latent representation of these encodings that you have, or and also like what was the intuition behind building up or having these computation models, and then why like are you comparing with the actual data?
Yeah. So for the decoding, the way we sell it is that so for example in neurons you want to see if there's like a kind of subspace of the neurons that can really explain your data. So we could do like more complex analysis from this, but we often don't have the data quality to do it.
So you're doing this trial by trial, or is it like you're averaging over all the trials by
No, we
segmenting
do
them
it
into fast, medium, and slow?
single trial.
Okay.
The unofficial reason that please don't tell anyone, but it's just that I'm an engineer and I like to have fun with decoders because it reminds me of my thesis. And
Glad to know, because even I was also working on the same pipeline. I just wanted to know your insights on the using these decoding networks.
and so yeah, there are fun challenges, and we have some, let's say, research access emerging where we want to go to newer technologies for psychiatry. So let's say all the know-how that we can build is nice to have. For the computational models, it's also because it's fun to do, but there's let's say a more serious reason is that we really want to tackle the underlying mechanism. And when you have these cognitive paradigms with a lot of conditions and a lot of correlated factors, I think it's often the best way to really understand what's let's say generating your data, because at some point you just get lost in different statistical tests otherwise.
So any comment on the heterogeneity of the data, because it is like the data has been recorded differently from each of the participants. It's not like generalized over all of them. So do you have anything to add on that?
Do you mean like the implantations are different across all the
Yeah,
patients?
yeah.
Yeah, well, that's kind of the little downside of these invasive electrophysiological studies is that we have no way to influence, and that's of course for the better, right? We don't want researchers to have any influence on the way these patients are taken care of. But then yeah, sometimes you get only patients with frontal implantations when you want actually to have the ventral temporal lobe. Then you're interested in the insula, but there are not enough patients. So yeah, you have to deal with the let's say most typical types of implantations. For example, the like occipital cortex is rarely implanted for epilepsy.
Okay, thank you.
Thank you so much for
Michael,
your.
I don't know if you can spend a couple of more minutes because there is one more question. If it's no problem, Etienne, you can go ahead.
Thanks for your questions.
Okay, hi Michael. Thank you for the talk. Very interesting. I'm happy that you go to recurrent networks, which are really working with animal models. So it's really interesting. Well, of course, you have much more data. I would like to know, because you say that you have untrained biological model. Does it mean that it is a naive model with no choice really of parameters, or it's just untrained connectivity? But I don't see really. Do you have enough data to really train the single synapses of this model? Or?
Yeah, so we were considering this. Actually, what we did is we took the Wang model. It's a spiking neural network for decision making, and Artemio kind of transformed the parameters from the Wang model to apply it to a rate-based model. Then, I mean, there are a few adjustments, like we try to adjust the decision threshold to get 50% hits and misses like in the data. So that would be like the adjustments that we are doing. Then initially we were thinking of training the model, and we had some ideas, but at the same time, even like on an expensive GPU, it does take like in the order of seconds to simulate a few trials, and so like applying some optimization methods is not at least yet feasible in the way that we implemented it.
Okay, nice. Thank you.
All right, thank you. I think there is no more questions, so time is up. So I guess we can wrap up here. Michael, once more, thank you for accepting our invitation. Thank you for this wonderful talk. And I really hope, I think everybody hopes that, since definitely our two groups have a lot of overlapping, I hope, this can be the pretext and inspiration for further collaborations.
Cool.
Good.
Yeah.
Thank you very much.
Thanks for inviting me.
Bye-bye.
Bye-bye.
Thank you.
Bye. Thank you.